So, it is wonderful to be here, and I'm very grateful to the organizers, especially to Peter Hedy, for putting this fantastic thing together. This is my second visit after 2017, where I had been with Bert Sakmann to one of the earlier events, and this is now even bigger and more fantastic here. I'm also particularly pleased that two of my academic mentors, Bert Sakmann and Erwin, are in the audience. It has been a long time that they've been listening to the simple things that we are doing in our lab. So this is the small institute in the old section of Munich near the English Garden, where we have our lab, and actually these two windows belong to Bert Sakmann's lab when he was at the Institute of Advanced Studies of our university, and this was part of our collaborative work that I in part will show here. The focus of my talk today is on the dysfunction of neurons and circuits in Alzheimer's disease. And in view of the large number of students, I allow myself to actually give you a little bit of basic background on this. So at the beginning of last century, Alzheimer, who at that time was a professor of psychiatry in Frankfurt, had this patient come to him, and he recognized that her memory is seriously impaired. If objects are shown to her, she names them correctly, but almost immediately afterwards, she is forgetting everything. So this was a relatively young woman, and that it was surprising that she had the signs of dementia that early. Then he actually, Alzheimer moved to Munich, became also professor of pathology, and then he looked at her brain in pathological sections and found postmortem showed an evenly atrophic brain. Specimens show very striking changes in neurofibrils, and there are military forces which are caused by the position of a special substance in the brain. By now, we know that this atrophy is remarkable, and here you see atrophy so no atrophy in the control specimens, and then with mild cognitive impairments, there are already pronounced signs of atrophy which become even stronger. So this is something you can see with modern imaging techniques in patients. We know that neurofibrils are actually the tau protein inside cells, and amyloid plaques are now considered to be one of the central hallmarks of Alzheimer's disease. All this knowledge of the years led to what's known now as the Amoled Hypothesis, and this hypothesis states that A-beta is the main cause of Alzheimer's disease. Other pathologic changes are downstream of A-beta accumulation. In Alzheimer's disease patients, the increased production or the decreased clearance of A-beta causes its accumulation in the brain. Soluble A-beta aggregates are the main pathogenetic factor, and I'll come back to that. But this entire hypothesis is still quite controversial. There are still people who say, now, maybe the relation between A-beta and the disease is not so clear, because there is really no clear functional correlation between the amount of amyloid beta and the clinical symptoms. And that's a long discussion, and the dispute is unresolved. Again, more background information. So amyloid beta is produced from the APP, or the amyloid precursor protein that's inside cells. And this is released in an activity-dependent manner from neurons after cleaving off these blue residues here by base 1, or the beta secretase, or the gamma secretase. Once released as monomers, there are aggregations as oligomers, dimers, trimers, and small oligomers, and eventually large plaques are formed in the brain everywhere. Now, where do we stay with our understanding? So over the years, there had been a lot of accumulation of knowledge on the clinical symptoms, brain pathology, and a lot also in the last decades on the molecular mechanism. A big gap of knowledge were what happens really on neurons on the single cell level or single synapse level in the brain. And of course, it's not possible to study single cells easily in a human brain, even though we are going to have two talks today that deal with physiology of human tissue. Still, it's a major challenge to do single cell studies in human brain. And people typically rely on animal experiments for this sort of work. And one of the classical ways of studying cells was in brain slices, in tissue slices that you keep alive. However, in animal models, aged animal models like Alzheimer mice, it's very difficult to sustain the tissue on a healthy level, and you can hardly trust what you're seeing there. Therefore, the real step forward came when it became possible sorry, one step back, when it became possible to do in vivo imaging experiments, calcium imaging experiments. And what we had developed almost 10 years ago was a way of staining neurons with calcium dyes. And in this case, you can resolve calcium changes in individual cells, and these calcium changes in the cell body of cells code linearly for the action potential activity in the cells. So this is a readout, and if you look then at sections, okay, so with this in vivo setting now, it was possible to compare wild-type animals and Alzheimer animals, and this is one mouse model that we had been using that had mutations in the APP and in presenilin, and these mutations actually lead to the formation of plaques. So early on, at 1.6 month of age, no plaques. Here you see at 9 months, lots of plaques. The plaques developed at the age of about 6 months in this animal model, and that's the time window where actually we did experiments. And we started out with a very simple question, asking ourselves, what happens in neurons that are near the plaques? Is there anything special? And we had a clear hypothesis based on what we believe to be the standard literature, namely, we expected cells to have no function to be decreased, and this decrease, dampening of function should be then maybe a readout of the impaired cognition. So we started out the experiment by doing activity measurements in the upper cortical layers, and what you record there is low frequency activity now as calcium signals here. When we moved to the Alzheimer mice, we found indeed that there were a fraction of cells that we call silent cells that showed no activity at all for the entire time of recording. Some cells in gray here were similar to the other ones, we call them normal cells. But then, and this was the unexpected observation, we found a fraction of cells that were much more active than expected. We call them hyperactive cells. And they became then really the focus of research, and I'm mostly going to talk today about hyperactivity. So if you now look at fields of views, then you see in the Alzheimer model many cells here in red that have large levels of electrical activity. And this is really strikingly different than in a control mice. In addition, and that is really remarkable, it's a remarkable example of translational medicine, if you like. So there had been the observation, first in mice, that a fraction of these Alzheimer mice show epileptic spikes. And this has been observed later on also in humans, and actually there's now a specific treatment for the epilepsy behavior that is in humans. And that is specifically for the type of epilepsy occurring in Alzheimer patients. So the summary of that work is that instead of the expected generalized dismantling of excitatory transmission, we found a combination of neural silencing and pronounced hyperactivity. Neural silencing and hyperactivity, as you could show, are due to an impaired balance between excitation and inhibition. And there was a preferential localization of hyperactive neurons near plaques. And as mentioned, we found this evidence for epileptic behavior in this fraction of animals. This observation of hyperactivity seems not to be restricted to animals, because studies in human patients using functional MRI show that especially during stages of early cognitive impairments, mild cognitive impairments, preceding the Alzheimer's disease, there's a significant increase in global activity, which is different from the single-cell activity that we measure, but it's still a readout of increased levels of activity that is followed at later stages of disease by a strong dampening. That is what we had actually expected to see. I'm going to jump over some levels of the study and would come to one of the studies that we did in collaboration with Bert Sakmann, where we asked the question of what comes first, silent or hyperactive neurons? And also wanted to know whether plaques are important for neural dysfunction. At that time, there had been a debate in the field. So some people working in the cortex considered that plaques are the more important factors. People working in the hippocampus said, no, it's probably the soluble A-beta. So this was an open discussion. And what we found is actually that soluble A-beta is the main player here. The way we addressed it was in many ways. I'm going to show you just one experiment. In this case, what we did was to take wild-type animals and APP animals at a stage where in this animal model there are no plaques yet. So everything that is there is a soluble part. Plaques are not really there. And it does not take really much analysis to tell that the level of activity in many of the cells here are by far higher than in the control mice. With this and several other experiments, we came to the conclusion that hyperactivity precedes plaque formation. It is the first functional alteration in hippocampal, but also in cortical neurons. Neuron silencing is a late event that seems to require the presence of plaques. Cellular beta is directly involved in the induction of hyperactivity, and the early hyperactivity is reversible and can be rescued by gamma secretase inhibitors. You remember from my slide, gamma secretase is important to cleave APP and to actually produce A beta. So with this knowledge and quite a number of other papers, we came to what we call now the hyperactivity trigger hypothesis, namely that the main factor in inducing the pathological changes is the early increase in soluble A beta. This leads to hyperactivity, and that can produce cognitive impairments in two ways: by a tonic sustained increase in activity that I showed you in the animals, but also by the epileptic one. This hyperactivity probably induces in these stressed cells that accumulate too much calcium changes that initially produce neurosilencing and probably eventually end up in cell death of these cells. It's an hypothesis that we still are working to improve. So this disturbed activity in neurons, of course, affects circuits, and I'm going to show you just one example which is in a way interesting because it relates to the disease. What we showed, what we looked at was a dysfunction of long-range cortical oscillations underlying sleep-dependent memory consolidations. So memory failure is one big issue in Alzheimer's disease, and one important way of consolidating memories are sleep waves, slow wave, waves in the range of 1 per second or so that occur regularly during a special form of sleep, the non-REM sleep, and we studied them in mouse models. And here you see, so we looked actually, we looked with a camera on the brain that was stained with a calcium dye. In the first trace, in these traces here, you show evidence that calcium and electrical recording show the same thing, basically. And if you look at activity in two spots that are far away in the posterior part and frontal part of the cortex, you can see that this wave superimposed almost perfectly. Yeah? So that this wave, so the entire cortex is in this up or down state, as you call it, if you record this electrically, during a sustained time, the entire non-REM sleep. If you do the same experiment in Alzheimer mouse, you see that there's a total disruption of this activity. The frequency goes up, and suddenly things don't coincide anymore. And the reason for this, and I'm not going to show you the evidence for it, but the reason for that is that instead of having a single pacemaker driving the entire activity, you have, like in heart failure problems, multiple pacemakers, and there's a desynchronization. And actually, this type of activity leads to very clear behavioral changes in mice. So mice that are trained in the so-called water maze, Morris water maze test, they cannot remember where the platform is. And interestingly, if you treat them, and I will allude to them immediately, then you can actually rescue. So you can predict from looking at the wave patterns, at the synchronicity of the waves in the front and back, whether the mice is going to behave behaviorally good or not. So we had discovered a severe breakdown of slow wave sleep oscillation caused by neuronal hyperactivity. The circuit dysfunction has direct impact on the consolidation of recent memories. And there's an effective rescue of both the sleep oscillations and memory defects by A beta scavenging treatments involving either, in this case, base 1 antagonists that we can apply, or the use of benzodiazepines that reduce neural excitability by enhancing GABAergic inhibition. It's a tricky thing. It cannot be easily done. It can be done in mice. In humans, I don't think it's going to work, but in mice, we can actually repair some of these Alzheimer's symptoms. Now, there is strong evidence also from humans for a disruption of the activity. The methods are not the same. So people use, in this case, PET, positron emission tomography, EEG, fMRI. But basically, they show then that the proportion of non-REM slow wave activity decreases dramatically as a function of increased A beta levels. And also the overnight memory retention decreased proportionally to that. So it's actually the same way of looking at things. It's actually a beautiful way of looking at this analysis because it's integrated over the whole brain and the experiments are relatively simple. Again, I'm going to jump over a number of studies and come to the most recent studies where we ask the question on the key mechanisms for a beta-dependent hyperactivation. What is it that causes? Is there a single mechanism or not, or how do we go about it? It was years of study. I'm just going to give you the bottom line here, and I follow Erwin Neher's work on synaptic transmission. So again, a synapse scheme. So in this study, what we found basically, or what we looked at eventually, was we looked at glutamatergic synapses in cortical neurons. So you probably all know that this synapse is the dominating synapse for synaptic excitation in the mammalian cortex. And for the students, just to repeat, so what happens here is that electrical activity, action potential, calcium influx causes glutamate release. And what's important to remember is that the glutamate accumulation in the synaptic cleft goes up to very high levels. For a very short time, glutamate goes up from nearly 0 to almost a millimolar. It's enormous. And in order to maintain the possibility of the synapse to run higher frequencies, it's important to remove it very quickly. And this removal is done by glutamate pumps that are mostly located in astrocytes. So this glutamate removal actually is essential to maintain the integrity of the system. And what we show then is that what A-beta does, it blocks this uptake of glutamate into the synapse. And this is the cause for everything that happens afterwards, according to our data. So again, in a scheme, so increased levels of glutamate are produced in the presence of soluble oligomers, and this prevents then the reuptake of glutamate in the astrocytes. And this glutamate hanging around produces a depolarization. There are glutamate receptors, of course, also in the cell bodies and everywhere. So this glutamate hanging around and produces a depolarization. And this entire process leads to a kind of vicious cycle. So you start out with a baseline activity. If you block reuptake, you produce depolarization by the glutamate accumulation, you increase the activity, and the cycle repeats itself and boosts, and you add that to the hyperactivity that I've shown you before. So we conclude from the study then the most early defect in Alzheimer's disease is a synaptic dysfunction, actually. It's a dysfunction of glutamate reuptake. Everything else seems to be working properly. The primary target of A-beta dimers, which are the most neurotoxic amino species, are the glutamate transporters located in the astrocytes. A-beta-mediated hyperactivation is due to altered synaptic transmission involving defective reuptake. A vicious cycle of hyperactivation is relevant exclusively from already active neurons. So the activity of neuron is driven by glutamate. If there's no glutamate to drive the neurons, uptake blocker will do nothing. So only the active neurons actually are mostly affected, and this may be one of the reasons why the activity starts in the hippocampus, because the hippocampus is naturally a very active region that, where things are. The basic mechanisms seem to be the same everywhere in the brain. And the final comment is there that A-beta containing brain excites from human AD patients. So we had received this excite from a collaborating lab in Harvard, and it turns out that this brain excite is even more powerful than the synthetic A-beta dimers that we had in the lab. So, final slide with the general conclusions. So what we conclude from this work in mice is that neurons and circuits are functionally impaired already at early stages of Alzheimer's disease before the onset of overt symptoms of dementia. Initially in a small fraction, but this extends gradually. The most early dysfunctions are triggered by excessive levels of activity. And often asked question is, how helpful are mouse studies? So mouse models of A-beta have limitations related mostly to the short lifetime of mice, the lack of brain atrophy, and the uncontrolled expression of A-beta in many transgenic models. So this is well known. We would argue, however, that crucial neuronal impairments are remarkably well conserved in mice. This includes the sustained hyperactivity, the susceptibility for epilepsy, impairment of the circuits underlying brain oscillations, and memory consolidation. Therefore, studies in mouse models remain indispensable in our view, and particularly useful for the analysis of early neural dysfunctions on single-cell studies. And we are convinced that concerted studies of humans and appropriately tailored mouse models can have a strong potential to clarify the mechanism needed for designing efficient treatments. I stop here with the list of people who contributed, and special thanks also to Bert Sakmann, our guest of honor today.
So, Professor Konnerth, thank you very much for the excellent talk. Unfortunately, in this session, there is no time for asking questions. But before you leave, we would like to give you the certificate and the medal.